Hi everyone, my name is Joanna Castillo-Hanes, and today I'll be presenting on cell biology of viral infections, focusing on the adenovirus. We'll walk step by step through how these viruses enter, traffic, replicate, and how we've even learned to repurpose them in medicine today. Adenoviruses are double-stranded DNA viruses that infect humans, can also affect other things like birds as well, but I'm focusing on the human part of this. They're responsible for respiratory illnesses, eye infections, and gastrointestinal issues. What makes them especially interesting in cell biology is that the same virus that can cause disease are also repurposed as vectors for vaccines and gene therapy. So studying adenoviruses gives us both infection mechanisms and real-world applications. But not only the main reason why I chose this topic or this particular virus was because this virus hits close to home to me. In October of 2019, my daughter, who was 7 at the time, was hospitalized with the adenovirus. It hit her gastrointestinal system pretty hard. She had high fevers, severe diarrhea. We really couldn't keep her hydrated, no matter the Pedialyte, the popsicles, everything we tried. She ended up being admitted in the hospital. She had IV antibiotics, IV fluids going in her for a few days, trying to control her diarrhea because it was pretty bad. It was a scary time for me. It was very scary for any mother to see their child in a position where my daughter was. And at that point, I really didn't know what the adenovirus was or how scary it could be for children or for anyone in the matter. Luckily, she did recover pretty well, though she still has some lingering GI issues that may or may not be connected to the virus, but that experience alone showed me firsthand why it's important to understand the cell biology of viral infections. Because once you know how they work and you realize how much impact they can have and why research into them is pretty important. Alright, so now let's talk behind the virus, the structure and its spread. The adenovirus belongs to the family called the Adenoviridae, with more than 50 human serotypes. Structurally, they're non-envelope viruses with an icosahedral capsid surrounding a double-stranded DNA genome. Replication usually occurs inside the nucleus of the host cell, which means they directly interact with our DNA machinery. Transmission happens through droplets, fecal-oral contact, or even eye exposure. While infections are often mild but can be severe, adenoviruses are a key model in cell biology because they manipulate almost every stage of the host cell's environment, from entry and trafficking to replication and assembly. Now I'm going to explain the entry point, where the fiber knob meets CAR. The first step in the adenovirus infection, is attachment. The virus uses its fiber knob proteins to bind to the CAR receptor on host cells. So think of it like a lock and key, where the CAR receptor is the lock and the fiber knob is the key. A specific region called the AB loop determines how strong that fit is. Once the virus locks in, a stable complex forms, and that triggers the infection process. So this lock and key step is central to cell biology because it shows how viruses rely on specific molecular interactions, and is also why this stage is such a good target for antivirals or for engineering of new viral vectors. Alright, so now we're going to move forward to trafficking and escape, hijacking the cell's highways. So once this virus is inside, it isn't just free floating. It's taken into endosomes, which are little compartments that usually break things down. but the adenovirus hijacks the system. They rely on host proteins like Rab7 and Rab11, which normally act like traffic directors to guide the endosomes. If these proteins are disrupted, the virus gets stuck and can't encode or release its genome. What's amazing is that the adenovirus even trigger signal cascades to remodel the endosomes, basically punching their way out. This shows how viruses exploit the cell's transport machinery for their own survival. Now for the viral proteins in action. Inside the nucleus, viral proteins take control. The 100K protein works as a chaperone, binding to hexons, keeping them from clumping and guiding them into stable trimers to form the viral capsid. Without it, the virus couldn't assemble properly. Another key protein, protein 7, undergoes post-translational modification that regulate how the viral genome interacts with the host chromatin. This affects nuclear import, gene expression, and genome accessibility. Together, proteins like 100K and 7 fine-tune the virus's ability to replicate by hijacking the host cell's nuclear biology. Now let's talk from infection to innovation. What is fascinating is how all this cell biology is repurposed in medicine. Adenovirus-based vaccines like the AD26CoV2S, CanSino's AD5, and Sputnik-like deliver the spike protein gene into host cells. This cell's own machinery then transcribes and translates that gene, producing spike proteins, which is recognized by the immune system. That triggers both antibody and T cells responses. Adenovirus vaccines are stable in refrigeration, highly immunogenic, and versatile. Beyond COVID, adenoviruses are also used in gene therapy, delivering therapeutic genes directly to patient cells. This is cell biology applied to real-world innovation, which is really cool in my opinion. All right, time to wrap it up. Adenoviruses show us how viruses hijack the cells at every step, from entry and trafficking to replication and assembly. Proteins like 100K and 7 fine-tune these processes, but instead of only seeing them as pathogens, we now harness them for vaccines and therapies. This journey from infection to innovation highlights why adenoviruses are such an important model system. They connect the details of cell biology directly to medical breakthroughs. Thank you.